Yeah, good morning, everyone. Today's lecture topic is transport of oxygen. I am Dr. M.I.M. Shuja, lecturer from Department of Physiology, Respiratory University of Sri Lanka. As you can see in this picture, the red blood cells are carrying oxygen. So the oxygen is transported throughout our body, mainly via conjunction with the some technical problem. The oxygen is carried from the body joining or combining with red blood cell. So the transport of oxygen is mainly considered when we are inhaling the inspired air is having oxygen. That oxygen passes through the trachea, goes into the lung, mainly the alveoli. From the alveoli, it diffuses through alveolar capillary membrane into the pulmonary circulation. In the pulmonary circulation, there's an equilibrium between oxygen and carbon dioxide. So more oxygen reaching the pulmonary circulation and deoxygenated hemoglobin or RBC become oxygenated. Then this oxygen carries throughout our body via systemic circulation and supply oxygen to the needy tissues. This is whole transport of oxygen from the atmosphere till the mitochondria in the tissue. We are going to learn how it happens. This is the objective, you can go through that. Oxygen delivery. The volume of oxygen delivered to systemic vascular bed per minute. The oxygen delivery depends on the amount of oxygen entering the lungs, adequacy of the gas exchange, and enough blood flow to carry the oxygen to the tissue. And another one thing is capacity of the blood to carry oxygen. That depends on the amount of dissolved oxygen, amount of hemoglobin in the blood, and the affinity of oxygen for hemoglobin. Affinity is going towards something towards like. So the dry inspired air, humidified air from the alveoli, it comes to the pulmonary circulation and systemic circulation. Oxygen cascade. I think I have taught you in my last lecture. The oxygen moves down the concentration gradient from relatively high levels in the air to low levels in the mitochondria. We call that oxygen cascade. So this is the thing. This is the atmospheric. Is a gas in the alveoli. Because of the diffusion, the capillary a bit low oxygen is coming. Because of the shunt, it is further reduced. So this is cascade. And I have explained it in the last lecture. So there are a few key steps in oxygen cascade. Uptake into the lungs, carrying capacity of the blood, delivery to the capillaries, from the capillary delivery to the interstitium, then they deliver to the individual cells, and the oxygen is utilized by the cell mitochondria. What are the determinants of partial pressure of oxygen? So this is in the artery. The arterial pressure of oxygen. The inspired oxygen concentration, the alveolar ventilation, VQ mismatch, and the diffusion of oxygen through the alveolar capillary membrane. These are the determinants which decide the partial pressure of oxygen in the arteries or capillaries. Oxygen and carbon dioxide transport depend on diffusion of gas and flow of the blood. The gases has to diffuse, the diffused one has to carry from the pulmonary circulation. They say presence of hemoglobin in blood increase oxygen carrying capacity by 70 folds. The chemical reaction of carbon dioxide, that is carbon dioxide react with water and form bicarbonate and go on. So that chemical reaction and carbon dioxide sometimes bound with hemoglobin and form carboxyhemoglobin. So those reactions increase blood carbon dioxide content by 17 folds. So in the alveolar, your partial pressure of oxygen is 104 millimeter mercury. From the pulmonary capillaries, that is from the pulmonary, that means the venous blood, which is coming via pulmonary capillary, that contains 40 millimeter mercury. So there are going to be equilibrium between oxygen in the alveoli and hemoglobin in the pulmonary circulation. They are going to get equilibrium. So this will happen 1/3 of the way through the pulmonary capillaries. That I think last time I explained what is perfusion limited. Okay. So this is the graph which states the perfusion limited of gas, and one of the example is normal oxygen diffusion in the normal condition. So if you see this is the pulmonary circulation, this is the distance of the pulmonary capillary, and by 1/3 of the distance it travels, it achieves the equilibrium. So this is again, I explained, pulmonary capillaries have more oxygen. They are mixed with shunted blood, that is deoxygenated blood, and there is a smaller reduction in the partial pressure of oxygen in the systemic arterial blood. So the transport of oxygen in the blood, it depends on rate of blood flow and the rate of tissue oxygen consumption. So in the artery, the partial pressure of oxygen is 95. Then it gets utilized by the tissue, and at the veins it is 40. The transport of oxygen in blood. Each 100 milliliter of blood is transporting 20 milliliter of oxygen. 97% of oxygen is transported in combined with hemoglobin. The rest of the 3% is dissolved. At high partial pressure of oxygen, oxygen binds more to hemoglobin. At low partial pressure of oxygen, oxygen released from hemoglobin. Transport of oxygen, there are If you can see, yeah, this is my little one. Okay, she loves me so much, so she always come and be with me. Okay, the transport of oxygen, this is just a summary. The 97% is bound with hemoglobin, that is chemical combination, and 3% dissolved in plasma, that is physiological combination. Sorry, physical combination. When hemoglobin is bind with oxygen, we call it oxyhemoglobin. Oxygen binds loosely and it is a reversible procedure. Because if oxygen binds too tightly, we cannot deliver that. So the binding is a kind of loose and it is reversible. Each molecule of heme carry 4 molecules of oxygen. And now we are going to look at the hemoglobin. Hemoglobin, by now I think from the biochemistry you should know. But anyhow we go through, is a protein. So what protein are made of? Polypeptide. So each hemoglobin is made of 4 subunits, 2 alpha and 2 beta. I am talking about the normal adult hemoglobin. Each subunit contains one heme moiety. Each heme is having a porphyrin ring complex that includes one atom of ferrous ion. So each hemoglobin is having 4 subunits. That means 4 heme. That means 4 ferrous ion. Each of the 4 iron atoms in hemoglobin reversibly binds one oxygen molecule. That is we call oxygenation. So the hemoglobin bind with oxygen, one molecule Hb4O2, then second, third, fourth molecule Hb4O8. These Hb4 mentioned for heme. It is a quaternary structure, so that is determine its affinity for oxygen. The hemoglobin in the deoxygenated form, that means when there is no oxygen, that is found in the tense configuration. At that point, there's a reduced oxygen affinity. But when one oxygen molecule is bound, then the configuration change. It becomes relaxed configuration, that is R. That has more affinity for oxygen. If you see, here this is a deoxygenated hemoglobin or deoxyhemoglobin. This is the heme, the iron. So this is the oxygen binding site. It is very tight. Here if you see oxyhemoglobin, the oxygen binding site is relaxed. Okay. So the amount of oxygen carried in the blood is oxyhemoglobin depends on the amount of hemoglobin present. Usually in adult, our hemoglobin level is 14 to 16 milligram per deciliter. Each gram of hemoglobin will carry 1.34 milliliter of oxygen. So total 15 will carry 20.1 milliliter of oxygen per 100 ml of blood. You should know these values. And whenever the partial pressure of oxygen is high, it binds with hemoglobin. So there is a greater hemoglobin saturation that happens at pulmonary capillary. When there's a low oxygen concentration or partial pressure, especially at the tissue capillaries, these oxygen from the hemoglobin is released. So there is a lesser hemoglobin saturation. So there are a few terms you have to know. What is oxygen content? What is oxygen binding capacity? What is percentage saturation? Like that, you have to know. I have given the definition. Just go through. Now listen very carefully. This is very important. Usually we assume our blood is 100% saturated with oxygen. Each gram of hemoglobin carries 1.39 milliliter of oxygen. But blood normally contains inactive hemoglobin also. That means that hemoglobin will not carry any oxygen. So from the laboratory findings, they have find each gram of hemoglobin carries 1.34 milliliter of oxygen. So the oxygen content in hemoglobin is 100 milliliter contain 15 gram. So 15 into 1.34, 20.1 milliliter. But blood is only 97% saturated because 3% is shunted blood. So because the 20.1 ml is for 100% saturation oxygen. As in the blood, it is only 97%. 100 milliliter of blood contains 19.8 milliliter of oxygen. In that also, 19.5 bound to hemoglobin, 0.29 is in the solution. When the blood goes through the tissues, from each 100 milliliter of blood, 4.6 milliliter oxygen is extracted to the tissues in a normal resting condition. So at the venous level, you will get 15.2 milliliter of oxygen per 100 ml of blood. At the venous point, the saturation is 75%, the oxygen saturation. So roughly at rest, our body tissues consume 250 milliliter of oxygen per minute and produce 200 milliliter of carbon dioxide. So that we saw in the last lecture, the respiratory portion, 0.8. Just refresh your last lecture, okay? So during strenuous exercise, can you calculate the content of venous blood? During exercise, what will happen is oxygen uptake by the tissue increases threefold. So usually for 100 ml of blood, 4.6 milliliter oxygen is extracted. So if it is threefold, it will become 13.8 milliliter per oxygen. So in the artery, you are having 19.8 milliliter. So the tissue extract 13.8. So the rest is 60 milliliter of oxygen is found in the venous blood. So this is again just a summary. Oxygen transport mainly by red blood cell and dissolved oxygen in the plasma. By red blood cells, 97% bound to hemoglobin, dissolved is other 3%. So dissolved oxygen obeys Henry's law. What is Henry's law? Is the increased partial pressure of any gas is proportional to the amount of gas dissolved in the blood or in solution. So this is again just a summary. The arterial blood oxygen saturation of the hemoglobin is 97%. Venous blood 75%. But partial pressure of oxygen is at arterial level 95, venous level 40. Then again comes the oxygen hemoglobin dissociation curve. Again, people think it is a difficult concept. No. As I say, it is also one of the very easy concept if you understand the basic clear. So, what is oxygen hemoglobin dissociation curve? If we ask in the question, you have to say it relates percentage saturation of oxygen carrying power of hemoglobin to partial pressure of oxygen. So it is hemoglobin saturation versus partial pressure of oxygen. If you plot in a graph, it will have a sigmoid shape because the hemoglobin is having T form and R form. Because of that characteristic T and R configuration, you will be having characteristic sigmoid shape curve. Combination of first heme in the hemoglobin with oxygen increases the affinity for the second heme for oxygen. The combination of second heme in hemoglobin with oxygen increases the affinity for the third heme for oxygen. Again, combination of the third heme in hemoglobin molecule with oxygen increases affinity of the fourth heme for oxygen. So the affinity of the hemoglobin for the fourth oxygen molecule is many times higher than that of the first oxygen molecule. That we call cooperative binding. So this is the graph. You should know how to draw a graph. So what is the easy way to draw a graph? You have to remember few points. If you can remember few points, especially 2 points, what is the arterial level, what is the venous level, then it is easy to draw. Usually, what is the saturation in the arterial level? We can assume that is, okay, tell me, usually, what is the partial pressure of oxygen in the artery? That is 100. So at 100, the hemoglobin saturation is 97.5. So you have put 1 mark here. Then what is the partial pressure of oxygen in the venous level? That is 40. So 40, hemoglobin saturation is 75%. So 40, 75. We again put another dot. Then we will see what about 50%. So 50, here they give 57, 50 is around 27. So 27, 50, you put another dot. Then you connect all 3 dots. You will get a sigmoid shape curve. This is hemoglobin. This is for myoglobin. We just concentrate on hemoglobin curve. So this is what I said. Arterial level, PO2 is 100. Saturation is 97.5. Venous level, PO2 is 40. Saturation is around 75. Then for the 50, you will get 27, something like that. You have to practice, draw the graph. When we asked you to draw the graph, you have to draw x-axis, y-axis, you have to name it, you have to label it. You have to label both x-axis and y-axis. That is very important. Labeling is very important. Then oxygen dissociation curve. The amount of oxygen that is saturated on the hemoglobin is mainly depend on amount dissolved. Amount of oxygen carried by hemoglobin rises rapidly up to partial pressure of oxygen 60 millimeter mercury. But above that, the curve becomes flatter. You see, above up to 60, the curve is rising. Beyond 60, the curve becomes flatter. So in this, even though you increase the partial pressure of oxygen from 60 to 100, the saturation of the hemoglobin increase from 90 to 100. When hemoglobin takes up small amount of oxygen, it converts from tense state to relaxed state. So relaxed state favors additional uptake of oxygen. Because of this cooperative binding, you will get a sigmoid shaped curve. This curve facilitates oxygen loading at the lungs and oxygen unloading at the tissue. So before going into this, we'll just think. I said we need oxygen for our body's metabolic needs. But the oxygen is carried in the blood via bind to hemoglobin. But we get oxygen from the lungs. So at the lungs level, the hemoglobin goes and attract more oxygen. It should catch more oxygen. Then it goes to the tissue level and it will give oxygen. That should be the normal way, right? That's how things happening. When you are going to get oxygen at the lung level, we call it loading of oxygen. When tissue level, when you are giving oxygen, we call it unloading or dissociation. These are the terms. Okay? Just understand the basics. Again, I am repeating. The purpose of the hemoglobin is to carry oxygen. So it has to catch oxygen at the lung level, release oxygen at the tissue level. Then only tissue will survive. So here, this plateau phase, the hemoglobin is saturated with oxygen. So when tense state change into relaxed state, what will happen? It will favor further loading of the oxygen. Here, drop. What will happen? When there is dropping, the oxygen is giving to the cell. So steep portion of the curve, steep portion, the dissociation portion, between 10 and 60 millimeter mercury, small increase in partial pressure of oxygen yield large increases in saturation of the hemoglobin. At the tissue capillary, blood comes in contact with the reduced tissue oxygen, and oxygen released from the capillaries into cells. That's why the hemoglobin saturation is reducing, because it reduces the oxygen. In the flood portion of the curve, it is we call association portion, greater than 60 millimeter. Large increases in partial pressure of oxygen yield small increase in saturation. At the pulmonary capillary, blood comes in contact with increased alveolar partial pressure of oxygen, and oxygen diffuses from the alveolus to the capillary. As the partial pressure of oxygen rises, oxygen binds with hemoglobin. So increases saturation of oxygen. Okay. Hope now you understand what is oxygen dissociation curve. You see, it is very easy to understand. You just remember the value to draw the graph. Again, another interesting concept is P50. P50 is the partial pressure of oxygen in the blood at which the hemoglobin is 50% saturated. Or in other words, when hemoglobin becomes 50% saturated, what is the partial pressure of oxygen in the blood? Usually that is 26.7 millimeter mercury. We call it around 27. It is a conventional measure of hemoglobin affinity for oxygen. Now we will combine the P50 with affinity. Just imagine if you are loving towards the hemoglobin, at low pressure you will go and bind. Okay? You got my point. If your hemoglobin is 50% saturated in, imagine in one incident, one hemoglobin become 50% saturated when the partial pressure of oxygen is 20. Another instant, another hemoglobin become 50% saturated when the partial pressure of oxygen is 40. So which hemoglobin is have more affinity? The hemoglobin which becomes saturated when there is low partial pressure of oxygen, that has more affinity. The higher the P50, the lower the affinity. The lower the P50, the higher the affinity. Okay? This affinity of hemoglobin for oxygen. As P50 increases or decreases, we say the curve has shifted. When the P50 is less than 20, that means higher the affinity is shift to the left. When P50 is greater than 27, that is lower affinity, is shift to the right. Now listen very carefully, this is an MCQ point. When P50 is less than 27, P50 is low. So affinity for the hemoglobin is high. So oxygen bound to the hemoglobin more. The release of oxygen is low. Okay. When P50 is greater than 27, the P50 is greater. So the affinity is low. So the hemoglobin bind less to oxygen. So the hemoglobin lose or dissociate or give more oxygen. At lung level, you should have higher affinity for oxygen to catch oxygen. At the tissue level, you should have low affinity for oxygen to release or give oxygen. You have to work on what I said for last 5 or 6 sentences. If you can work it out, if you can understand, you understand the oxygen dissociation curve. Then the exam question is going to be just a walk in the park. So just go through this. This is your normal curve. This is your left shift. What is left shift? Hemoglobin affinity for oxygen is increased. So the loading is increased. So that will happen in the lung level. This is right shift. What's happened? The hemoglobin affinity for oxygen is reduced. So oxygen is released in the tissue level. So there are few factors you have to remember. I will make you easier how to remember. So imagine you are doing an exercise. When you do exercise, what you need? You need more energy. To produce more energy, you need more oxygen. So at the tissue level, your hemoglobin will release more oxygen. If hemoglobin wants to release more oxygen, it should reduce its oxygen affinity, or it should reduce its affinity for oxygen. When the hemoglobin affinity for oxygen is reduced, the curve is going to shift to the right and the P50 is increased. Okay, that you know. So when there's an increase, when you are doing exercise, the curve is going to shift to the right. Now you are all okay with that. When you're doing exercise, what will happen? Your oxygen is used. Your carbon dioxide is produced. So your carbon dioxide level is increased. When the carbon dioxide level is increased, what will happen? It combines with water and produce H plus. What is H plus? When H plus is increased, what happens to the pH? The pH is reduced. When you are doing exercise, because of the muscle contraction, what will happen? The temperature is increased. When there is a more H plus, what will happen? There is acidity. Okay? When you are doing exercise, the 2-3 dBg also increases. So right shift occurs at acute acidosis, high carbon dioxide, increased temperature, high level of 2-3 dBg, and another one is abnormal hemoglobin. Now you see, do you need to by heart the factors which make the right shift of the oxygen dissociation curve? No, just work it out. Then it will be very easy for you. You don't need to by heart for the exam. Okay? So you just go through these. These are few examples for what are the right shift, what are the left shift. Again, factors affecting dissociation. I have given you example individually. What will happen in the blood temperature, blood pH, carbon dioxide concentration. Again, a nice summary. Just go through this. Then we come for the another fantastic or one of the marvelous physiology, Bohr effect. Again, people believe it is a difficult thing. No, it is again one of the easiest concepts. What is Bohr effect? The decrease in hemoglobin affinity for oxygen when carbon dioxide of blood rises. What will happen when the carbon dioxide is increased? For example, during exercise. So during exercise, you have to give more oxygen to the tissue. So your hemoglobin will release more oxygen. That means hemoglobin affinity for oxygen is reduced. That is right shift. So that they say in a different way. The decrease in hemoglobin affinity for oxygen when carbon dioxide pressure of the blood rises, because the H plus which is formed because of the increased carbon dioxide, binds this histidine residue in the hemoglobin and reduce its ability to bind oxygen. The pH of the blood falls as its carbon dioxide rises. It shifts the curve to the right. So when the carbon dioxide is increases, the curve is shifted to the right. Then the hemoglobin has a low affinity for oxygen. That is Bohr effect. When the carbon dioxide is low, other way occur, hemoglobin has high affinity for oxygen. This again we in this graph it shows how the reduction in the pH, that means increase in the acidity, causes right shift of the oxygen dissociation curve. So reducing the acidity means increase in the carbon dioxide, increase in the H plus. Okay. So not reducing the acidity, reducing the pH means increase in carbon dioxide, increase in H plus, increase in acidity. But when the acidity is increased, the pH is reduced. So when the acidity goes up, the rightward shift of the oxygen dissociation curve is occur. So this is what I have explained earlier. At the lung level, carbon dioxide move from blood to the alveoli. So at the pulmonary capillary, there's a decreased carbon dioxide. So the curve is shift to the left. Hemoglobin is having more affinity for oxygen. So it catch oxygen. At the tissue level, there's an increased carbon dioxide, the decreased affinity of hemoglobin for oxygen. So oxygen is released to the tissue level. Very simple. So what we saw earlier is the implication of the Bohr effect. We just go through this. And the double Bohr effect. It's again very fascinating. One of the wonderful thing in the mother-baby love. Same principle apply. Your baba needs oxygen from the mother. So mother has to give oxygen to the baby. Baby has to get oxygen. So the fetal blood will lose carbon dioxide. Carbon dioxide diffuse from the fetal blood to the maternal blood. So when carbon dioxide reduces in the fetal side, what will happen? Hemoglobin affinity for oxygen is increased. The left side shift of the ODC, fetal hemoglobin take more oxygen. Meanwhile, maternal blood will get carbon dioxide which came from the fetus. So, affinity of hemoglobin for oxygen is reduced. Right shift of the oxygen dissociation curve and increase oxygen to the increase oxygen from the maternal hemoglobin. So baby will get more oxygen. So this is we call double Bohr effect. Wonderful, isn't it? This physiology. You all should love physiology. Okay, this is very wonderful. You don't need to buy heart at all. Just know the basic concept, work it out. You are okay. So this is a fetal because the fetal has more affinity because it catch oxygen from the maternal hemoglobin. And 2,3-diphosphoglycerate or 2,3-DPG. It's formed in the RBC. It's an intermediary in the Ebden-Mayhoff's glycolytic pathway. I think you know better than me because you all study biochemistry. I don't. So it binds to one of the beta chain in the deoxyhemoglobin. An increase of the 2,3-DPG shift the curve to the right, so causing more oxygen to be liberated. So this is the role of 2,3-DPG. It's found in the RBC. So these are the few factors which increase level of 2,3-DPG. dBg, like cellular hypoxia, anemia, hypoxemia secondary to COPD, and ascending to the high altitude. There are few factors which reduce the level of 2,3 dBg: septic shock, acidemia, and stored blood. And here are few factors affecting concentration of 2,3 dBg. pH. Acidosis reduce the 2,3 dBg level. Thyroid hormones and growth hormones increase. Stored blood is deficient in 2,3 dBg. Exercise increases to 3 dBg, but that is for 60 minutes. High altitude increase to 3 dBg. And in anemia and hypoxia, you will get increased level of 2-3 dBg. In the stored blood, what will happen is the 2-3 dPg, the 2-3 bpg level is reduced. Sorry, this is 2-3. Some people say 2-3 biphosphoglycerate, some say di. Okay, both are acceptable. The 2-3 bpg level falls, and the ability of this blood to release oxygen is reduced. Because we saw when there is an increase to 3 dPg, it binds with the hemoglobin, it releases the oxygen. So the stored blood, the amount of 2-3 dPg levels are low, so the oxygen releasing affinity or the power is reduced. But if you store in citrate phosphate dextrose solution, then the acid citrate dextrose solution, it can be, the effect will be less. So what happens to the oxygen hemoglobin dissociation curve in anemia? In anemia, oxygen carrying capacity is reduced and oxygen content is reduced. Saturation remains normal. So is a normal ODC, oxygen hemoglobin dissociation curve. This is in anemia. The shape of the curve is almost same, but the saturation oxygen content is reduced. Saturation remains normal. Here, this is oxygen content in the y-axis. Okay, it's almost half. What will happen in carbon monoxide? Affinity of hemoglobin for carbon monoxide is 250-fold relatively high to the oxygen. So it's compete with oxygen binding, left shift. So it interfere with the oxygen unloading at tissue level. It causes severe tissue hypoxia. Because when it's come to the left shift, the affinity for oxygen is increased. So it will not release oxygen to the tissues. So your tissue will get low oxygen, that will lead to hypoxia. That will happen in carbon monoxide poisoning. This is the graph I have explained. The utilization coefficient is, it is the percentage of oxygen extracted by your tissue per 100 milliliter of blood. So usually 20 milliliter of oxygen is transported, 5 ml of oxygen is extracted. So utilization coefficient is 25%. But in exercises, it can increase up to 75 to 85% because more oxygen will be extracted. What happens to the oxygen hemoglobin dissociation curve in exercise? I have explained to you. You just go through. The diffusion capacity of the oxygen is increased, so more oxygen comes. Additional capillaries are open, improved BQ ratio. So when you are doing exercise, increased carbon dioxide release from exercise muscle, hydrogen ion increases, pH reduces, temperature increases, 2,3 dpg increases. So there's a right shift of the oxygen dissociation curve. So more oxygen is liberated to the tissues. These are just a summary of what I have been talking. And we'll see what is myoglobin. Myoglobin also iron containing pigment. It resembles hemoglobin. It's found in the muscle. Its one molecule or one mole is bind with one oxygen mole, not 4 moles of oxygen. So it has lack of cooperative binding. So the curve of the myoglobin is rectangular hyperbola. So this curve is shift to the left side. So when you shift the curve to the left side, what will happen? Hemoglobin affinity for oxygen is increase. So myoglobin will have more affinity for oxygen. So myoglobin is found in the muscle. The hemoglobin is found in the blood vessel. So muscle myoglobin will get oxygen from blood. So what will happen in carbon monoxide poisoning? I have you. Carbon monoxide is a odorless, colorless, non-irritating gas. It competes with oxygen for binding site on hemoglobin. So exposure to carbon monoxide reduces the oxygen binding to hemoglobin. Person with 50% carbon monoxide block of normal hemoglobin have a leftward shift of the oxy carbon monoxide hemoglobin curve. Because it shifted to the left, the remaining oxygen molecules have more affinity for oxygen and remaining hemoglobin molecules have more. So remaining oxygen molecules will have more affinity for hemoglobin molecule. So they are tightly bound with hemoglobin. So unloading of the oxygen is slow. When there is an unloading of oxygen is slow, what will happen is there is a hypoxia is occur. But the partial pressure of oxygen is normal. So there will not be feedback mechanism to increase your oxygen level. So the treatment is you have to give pure hyperbaric oxygen. Hyperbaric means more pressurized oxygen. So the diffusion of the oxygen in the blood is increases. So hypoxia will be settled. So this is the normal hemoglobin, 0 carbon monoxide hemoglobin. Here the percentage of carboxyhemoglobin is increases. So when the carboxyhemoglobin percentage increases, the left shift of the curve is increases. So the more affinity for oxygen, so it will not release the oxygen at the tissue level. So tissue will get hypoxia. Hope you understood this. This is again very easy concept. Again, methemoglobin is just an abnormal form of hemoglobin. Usually in the heme moiety of the hemoglobin, iron is found in the Fe2+, that is ferrous form, but here it is found in the Fe3+ ferric form. So it cannot transport oxygen or carbon dioxide. And your homework is you have to search and find effect on oxygen hemoglobin dissociation curve when there is a increased methemoglobin level. Okay. Usually 1% of methemoglobin is normal and methemoglobin has increased affinity for cyanide. So just search and found and try to draw this curve. You will get more understanding. And with that, we come to the end of the lecture. If you have any question, yes, I believe my wonderful students are very clever. They understood everything. If you have any question, you can send me email or contact me via my WhatsApp. As I always say, may peace be upon you. And thank you very much. Have a nice day.